Thanks.
Okay, lucky second last. I've really enjoyed hearing everybody's talk now the day. And yeah, I think this presentation is the most meta presentation I've ever done, right? Like I'm presenting here at this workshop about another workshop that I attended a couple of weeks ago in Canada about tree breeding resistance, which is not my field. So I'm just going to talk about some of the key learnings in this meta presentation of different workshops and some of the key recommendations for how we potentially further some of this resistance breeding work that has come up in several other presentations. So the way that this presentation is structured will quite neatly, bring together some of the themes that have come up, which is a nice coincidence. So, let's kick off by acknowledging that we're on the traditional Maori lands and also acknowledging the traditional owners of the lands on which I live and work, which is the Myrtle people, in particular the Gorong, and Water people. Paying respects to elders past, present and emerging. I'm going to talk really briefly about my group, which is the Dieback Working Group, just to give you some context about who I am, why I'm here, and why I'm talking about this. So we're a grassroots community organisation. We're a not-for-profit, founded in '96 and incorporated in 2014. We like to support community leadership in the environmental biosecurity and plant disease management space. So in that regard, we do a lot of training, a lot of education programs, but we also do a lot of advocacy and policy reform and SME kind of input into different plans and advisory groups. And on that note, I sit on the National Myrtle Rust Working Group along with several other people who are online and in this room, including Craig. We meet several times a year from all the different myrtle rust affected states to talk about the National Action Plan, how we're furthering the objectives of the plan, and what are some of the key actions that are currently happening and that need to happen in the myrtle rust conservation space. These are just the objects of association for what my organisation does, and you can see we primarily deal with Phytophthora, but also other plant diseases and pests of environmental significance. Okay, so I just want to point out in an Australian legislative context, we've currently got the Threatened Species Action Plan, which has had a very positive election result in terms of the environment and species conservation. And the Threatened Species Action Plan actually has a lot of references to myrtle rust and what a threat it is for pushing some of our species towards extinction. So the funding attached to that plan is what came through our federal government to send me to Canada and attend this workshop that I'm presenting on today, and the National Action Plan there is developed through ANPC and something worth knowing about. So I am going to talk a bit about what is resistant breeding. I need to be able to see my slides because this is not my area of expertise. So I've recently learned when we're talking about resistance, we're not talking about some kind of silver bullet. We're not talking about some magical solution here. We're talking about a mechanism, and it can be to any extent, which lessens or slows the impact of the pathogen in a host. So, it's not a complete elimination of disease symptoms. And I've included this on the right. This is blister rust over in the United States. I was at the Durena tree breeding facility in Oregon, and you can see there's you can see there's a really nice sort of expression of a canker there. This is a resistant variety of that pine. It's still showing disease symptoms, but it's really healthy. So we're not necessarily looking at zero rust. We're looking for that tree is persisting and thriving and hopefully reproducing even if the rust exists. I also wanted to just foreshadow what I'm going to talk about, which is, it's not a silver bullet. It's rarely a 100% fix, but it is a strong potential tool for conservation alongside others. And, as I said, I think we're going to be wrapping up, this presentation wraps up really nicely some of the other presentations and what we heard about from Anne, and, what we heard about in terms of microbiome from quite a few presenters, is there are multiple approaches that we can use to manage a threat, and tree breeding is just another arrow in our quiver, so to speak. So we're thinking about the fact that there are two different kinds of resistance. We've got major gene resistance and quantitative resistance, and I'll explain that in a little bit. And then also, you know, there are some approaches which include hybridization or grafting with closely related species. That was something that happened with the American chestnut, and hybridizing with the Chinese and Japanese chestnut varieties to try and, you know, preserve that species. But it's hard to predict the ecological impacts. So really, we're wanting to keep our species pure and not go down a hybridization kind of approach. So when we're thinking about that, resistance in a species may be extremely rare. You know, there may only be a handful of survivors after a pathogen comes through. But generally, from what I learned at this workshop, and the experts globally were very adamant, generally resistance does exist within a species. It just may take several generations of crossbreeding to try and tease out an amount of quantitative resistance. This is just an example of the major gene versus the quantitative or partial disease resistance, and I guess a word of caution here. So you can see over on the left, the yellow line shows the percentage of mortality for a particular family of trees which were resistant to blister rust. This is the Port Orford cedar. And you can see there was a 0% mortality with the original strain of the rust. And so that's really, really great. You've got absolutely no disease symptoms in these trees. They're doing fantastically. But then there was a virulent strain of the rust that arrived in North America. And it was different enough that the resistance that they had bred into the Port Orford cedar was then completely nullified by this virulent strain. And you can see the mortality rate of that family just, yeah, sorry, hang on, completely climbing in that context. So we need to think about, you know, if we do find major gene resistance, then it's fantastic. But if we can find that sort of partial resistance, which is where, you know, you still get symptoms, but the trees are still, you know, doing well enough, then that's something that we shouldn't underestimate as a tool. Not sure I did a great job of talking about that, but again, not my field. So really basically, what is resistance breeding? And, you know, very typically we see this resistance breeding project cycle. You have a healthy nuttery, you have a healthy forest, you start to observe decline due to a pest or pathogen. It can be introduced, or actually in an increasing number of cases, it can be native. And then, you know, people start to panic. We start to do things like collecting germplasm from what's termed the lingering individuals. You might have heard the term lingering ash. Those lingering individuals are then used in a breeding program where we're back crossing individual parent plants or families that are showing signs of resistance. And you can do this for several generations, and it should be an iterative process. As I said, it's not a silver bullet. It's not a straight up, you know, cure for something like myrtle rust. You then need to take those results and confirm or quantify the resistance in a series of nursery or field trials. This can be as complicated or as simple as your resourcing allows it to be. And there are a million different ways to test this kind of thing. And then, you know, ultimately, hopefully, you're having the production of disease-resistant families, which can be used in a conservation context or in a market context. And alongside that, we're looking at ongoing monitoring and research. But there are heaps and heaps of things that we need to think about when it comes to considerations and cautions. And I apologise for the horrendogram. This is one of the key sort of things that we think about in a plant pathology kind of field, where we're looking at the tree disease spiral and all the different things that can impact the health of our forests and our plants that aren't just a disease, but that make our forests less resilient to disease. So I guess considerations and cautions that sort of I picked up from this conference is, I'm going to come back to the top one, but the disease spiral spread principle still applies, right? So we still need to think about, even if we have a resistant species, we still need to think about that resistance might not persist if the plant is stressed because of abiotic factors like climate change. It might not persist if there's a particularly bad pest year, you know. That plant can still be extreme, can still be vulnerable if we've got these other factors creating lower resilience. For that reason, to have a successful resistance program, we need to think about factors like a resistance breeding program. We need to be thinking about factors like the ecology and biology of the pathogen and the host. So, you know, what kind of generation time do you have for your plants that you're trying to achieve resistance? How does that affect the feasibility of being able to create a resistant family? What's the potential differences in phenology or flowering time for different plants? So if our objective is to get a family of a particular species that can reproduce naturally in its own ecosystem with myrtle rust present, you know, we need to be thinking about, you know, potentially something that flowered earlier could time a little bit more resistant to them. So breeding for particular phenologies actually is a particularly common approach to tree breeding resistance. We need information on hazard modelling, so where are particular hazardous sites. We need to understand host population genetics. We need to understand seed zones. So it's complicated. We need to know about abiotic influences, and we also need to know about the management of other threats. And we need to be factoring that in. So if we go to create a seed orchard of, you know, a rare species for resistance breeding, we need to be fencing and making sure that we're not having predation by herbivores. And there was some really sad case studies that were talked about in Canada around deer and, you know, seed orchards with material of that had been sort of 50 years in the making, and it got decimated by deer. So management of other threats is very important. And we also need to think about the fact that there is the potential for pathogen adaptation. So, you know, we do go and breed a resistant variety, particularly if we go down the major gene resistant pathway. And then you have the pathogen evolve, or you have a new strain arrive, you're potentially back to square one. So I want to come back to the top, which is cultural considerations. It got touched on briefly at the conference in the co-op breeding program that's happening in Hawaii. So in many traditional cultures, it is taboo to move plants, even if they're the same species, across traditional boundaries. So I put across Budja because in my part of Australia, that's the Noongar term for country. But, you know, we need to be thinking about respecting cultural protocols when we're thinking about these kind of initiatives. And on that note, I just wanted to highlight something that we haven't raised, which is Indigenous cultural intellectual property and benefit sharing when it comes to the potential economic application of any successful breeding programs. So, you know, if it is a commercial species that we're looking at. And these are underpinned by the UN, and also I really recommend having a read of True Tracks Principles by Terry Janke if that's an area that you are interested in. I need to pick up the pace, I think. So obviously with myrtle rust, we're dealing with a very complex problem. People at the IUFRO workshop were captivated. They were like, that's so weird to have a pathogen that has such a massive host range, right? And so many potential ecological impacts. Where do you even start? How do we prioritize what we're going to do? You know, and the fact that it is an ecological problem as well, not necessarily a commercial problem, primarily, does make it more challenging in that regard. So, you know, that was sort of food for thought that I gave to people in conversations that I had at the conference. You know, in some cases we're racing against extinction. In many cases, we're not getting viable seeds. So it's a really difficult situation. And defining our priorities is a challenge. You know, speaking to Craig about whether or not we do focus on threatened species conservation as our priority, or do we focus on many of the people in the workshop straight away latched onto the fact that myrtle rust wasn't yet in some places in Australia. So do we then focus on preparedness and getting ahead of the curve and seeing what's susceptible? do we focus on preserving ecosystem function? You know, there are some, I put this picture here because I've heard just some crazy stories about the loss of these bank stabilisation species up in North Queensland, creating siltation of the rivers, creating sedimentation in the reefs, including the Great Barrier Reef. So, you know, these stories are incredibly complex, and we need to be thinking about this from a functional ecology perspective, as well as just from a pathogen management perspective. Do we think about culturally significant species and preserving things through that lens? Or, you know, is it an economic question? I would argue that we probably need to be thinking about all of these things at the same time and opportunistically, you know, pursuing projects as they arise. We don't even know when we're thinking about what resistance breeding program for Myrtle rust, what our potential objectives or definition of success is. It's complicated compared to a production focus, but what are we seeking to achieve? So, you know, success in a classic tree breeding sort of framework is really defined through the three factors here. We talk about durability. We need to make sure that the pathogen, well, basically that the resistance that we breed for isn't likely to then be nullified by another strain being introduced or, you know, the pathogen or pest evolving. So the work that Mike was doing, tracking whether or not we're seeing sexual reproduction in Myrtle rust, and the work that Ash is doing in ANU through constantly reevaluating whether or not those strains have different markers is really pivotal when it comes to durability. Stability, we need to be thinking about this over a range of climatic and environmental conditions. So several of the experiments that we talked about in the conference showed that the resistance genes were nullified if we saw a 1.5 degree temperature increase. And, you know, that 1.5 degree temperature increase sounds pretty familiar at the moment. So we need to be thinking about making sure that we're running those experiments to see if we are looking for resistance, and then we test those seedlings in, they just had them on heating pads in the glass house. Are they still resistant? And then usability, like what percentage does suit our needs and objectives from a conservation perspective? And how can we use other management tools like potentially DSRNA or fungicides to complement a certain percentage of resistance, maybe in a bad year? We talked about having a couple of back-to-back linear years. You know, potentially you can employ other management tools if you know that these plants are likely to be more vulnerable in that year. My mission, I felt like an investigative journalist. My mission was basically to find out what do we need to get something like this off the ground and write it into a report for the Australian federal government. And visiting Dorina in Oregon, which is one of the sort of world-leading tree breeding for disease resistance facilities, was a real eye-opener in terms of, like, it doesn't just take a few resistance breeding experts and a few glass houses. Like, it takes a humongous amount of effort, even in terms of just people mowing the lawn, people fixing the retic, the amount of man-hours that it takes to do the annual surveys of, you know, the canker symptoms, the blister rust symptoms, to the extent that was needed for the statistical analysis was significant. So, you know, thinking about facilities, thinking about people power and, you know, human resourcing, and thinking about funding are obviously barriers to this, but I don't think that we should necessarily let them get in our way. Like, it seems like a big thing, but we do have, well, I'm going to get onto this in a few slides' time, but, you know, in the examples that I saw, these collaborations and using facilities across different organisations between industry and government and community were key to ever making this work. Which I think is a very fitting sort of thing to think about at this event. And then time. So a lot of the resistance breeding projects that I've heard about to date have been associated with PhD projects, which are amazing. I'm a huge fan of student research and early research projects. I think you get some incredible results, but, you know, trees have a very, even to produce a disease-resistant crop cultivar is 18 years on average, right? So from a tree perspective, we need time, and we can't be limiting ourselves to 3-year election cycles or 3-year funding cycles. But how do we address that? So many other things to think about, guys. Surveillance is key. So there were a lot of citizen science approaches that we used to try and find, when I'm talking about surveillance, I mean, you've got a species that is at risk, but how do you know which plants to start from in terms of breeding? Like, where do you collect your seed from in the first place? So citizen science looking for resistance can be really important, but something that came up at the conference was, yeah, one of the approaches that we're currently using in Australia is completely novel, but yeah, I'll get into that in another slide. You need hazard mapping, so you need to be able to understand like where your disease risk is and your site risk ratings, and you need some defined seed zones, and these are usually defined per species. So you need to understand your population genetics for that species, your climate, your elevation, your soil type. Cultural protocols need to be taken into consideration if there are those taboos around moving plants across cultural borders, and then other threats like herbivory and so on. And then ecological niche preservation is an interesting one. If you have a species, I'm not sure what's happening in some of the ecosystems that were formerly dominated by redundant vessels, but you know, you've got this communities that are dominated by a species that suddenly starts to die out extremely quickly because of a pathogen. How do you preserve those sites to then potentially reintroduce resistant stock at a later date? Because you know, you see a lot of weed invasion and all that sort of stuff, or natural succession of native species. So I want to end on a high note because I've just told you all about how complicated it is. We already actually have quite a lot of stuff going on, which I think we all know because we're at the end of 2 fantastic days of hearing about all of the things that we have going on. But, you know, we have active monitoring and germplasm collection programs, and the sort of novel surveillance that I was talking about earlier really comes out of a project that's led through Queensland Department of Agriculture and Forestry, and I think still funded through the IUFRO office, yep, which is the Australian Chief Environmental Biosecurity Office. And that is working with Indigenous ranger teams to not only surveil for myrtle rust symptoms, but also surveil for myrtle rust resistance, and then having an ongoing GIS database. And that's a program that's been actively happening for a few years now. And, you know, it wasn't something that people had heard of happening anywhere else in the world from a resistance breeding perspective. And it's got us a head and shoulders above what we should be looking for in terms of where we source our seed from and our germplasm from. Existing ex situ populations for some species exist, as we've just heard about through Craig and others. And we have really well-researched protocols for tissue culture and seed storage, which again puts us ahead of the curve for anything like this. You know, if we have those tools down pat, then we can relax a tiny bit because we've got our insurance populations. We've got really strong collaborations and networks. We've got excellent existing climate models and disease risk models. We've got an excellent global research community, including, you know, some really cool findings on accelerating the generation time for mutation species coming out of South Africa. You know, applying certain chemicals to be able to get seed from plants quicker. Also, fantastic research on how we can use fields like metabolomics and microbiome studies to enhance resistance. So, for example, spraying methylplasminate, you know, that's been shown to be effective in producing a resistance response in trees that in plants that don't have that genetic resistance. Anyway, 2 minutes. And then we've also got a range of awareness raising tools. And I'm doing a shameless plug here for my organisation. We, in Western Australia, run something called the GWG Green Card training, which we then use to raise awareness about environmental biosecurity threats, including myrtle rust, because in Western Australia we don't have it. And so upskilling people to be able to surveil for it is super important. So these are just a few slides to give you some homework should you be interested. If people want to take a photo of this slide, this is 2 documentaries created in relation to the Indigenous Ranger surveillance programs that I was talking about. So they're really great little resources, and very sad and uplifting at the same time. This top one was showed at the last conference when Jeff Pegg and AJ Perkins and Tilly Davis just launched it, but this bottom one I don't think has been shown at this Australasian Symposium yet. This is the plug about the green card training. So this is the kind of content that I deliver, or myself and my trainers deliver when we talk about nodal rust in Western Australia. So, you know, we'll show people the typical symptoms, photos of, obviously, we can't take them out in the field and show them. I love this little demonstration of all the different things to look for. The training is primarily around phytophthora and soil-borne pathogen hygiene, but you know it can be very nicely adapted to biosecurity threats. And I just wanted to do a little shout out to the fact that we're currently investigating the feasibility of a green card Aotearoa to expand into working with kauri dieback and very basic hygiene training for people who work in the kauri forest. And yeah, we will be including myrtle rust content in there for some basic awareness training. It's a very low level kind of course that takes people from zero literacy to basic literacy pretty much. And another thing that we do have that we need to make the most of is, you know, we've got some really solid case studies of success stories around the world of where this resistance breeding has worked. So, you know, funding wise, yes, it would be good to have a proof of concept which is Australia specific or New Zealand specific, but the fact that we have these examples like the white pine globally to be able to show that this stuff does work is a real good starting point when it comes to our value proposition. So, concluding remarks, I'll be writing all of this up into a report. I'm sorry this presentation has been rushed, but I literally just got back from America yesterday, so I didn't have much time to pull this together. And we'll have key recommendations and next steps. So I'm sure that will be made available. I can email Jenny for distribution once it's ready, which will be in the new financial year. I'm really recommending we engage with international experts and then local experts as well. There are people who do resistance breeding in Australia and New Zealand. It's just there. You know, generally not working in a conservation focused area. So it's very interesting to have those global experts. And we do have an opportunity to host the next one of these true resistance breeding workshops in 2027. People are very keen to do the first ever Southern Hemisphere version. So watch this space. Maybe Jenny will get bored now that she's finished. I'm going to be recommending we really need, considering the size of the problem, we need some kind of framework for prioritisation of species because, you know, where do you start? It's not good. And we need an integrated planning and management approach that talks about all of these different tools. I'll leave it there. I was going to talk about those photos, but I think I'm out of time. So thank you. Thank you.